Good afternoon, dear students. Today, Department of Histology and Embryology represented for you the lecture Connective Tissues. The famous American writer Dorothy Parker was writing many years ago that the beauty is only skin deep, but ugly goes clean to the bone. So this is a very interesting phrase to reflect it through this structure of connective tissue. As a connective tissue of skin, we have seen here dermis and epidermis, like epithelial tissue. This is a dermis. Also play the key role in the process of senescence. The blood capillaries, which is a lot of housing this area, to play the key role also in all types of connective tissue. Because that this beautiful young girl face doesn't have any wrinkles, as opposite that the face and skin of old man is a lot of wrinkles can be seen. Ilya Mechnikov, famous Russian biologist and histologist, was investigate the senescence of human organism, and of course you know that Mechnikov is the author of phagocytosis theory and theory of cell-dependent immunity. But during his investigations of senescence, he have to describe that the person is as old as his connective tissue. So connective tissue reflected all changes in human body because it's represented in each region of human bodies. Also very important to know the definition of connective tissue. So let's now we're ready for writing. This is definition of connective tissue. Let's now write that connective tissue is a complex of mesenchyme derivatives consisting of cells differons and big quantity of extracellular matrix, fibers and ground substance, participating in supporting homeostasis of a body internal environment and differing from other tissues by lesser need in aerobic oxidation processes. Of course, this very difficult definition for immediately answer, but we just step by step to have memorized, these important terminological moments. First of all, connective tissue is a complex of mesenchyme derivatives. Of course, mesenchyme, like embryonic connective tissue, give rise to formation not only connective tissue but also vessels, smooth muscles. But everywhere where we have find the vessels we have to see in the connective tissue, different types. Because that we also seen that cells differons, connective tissue consisting of cells differons and a big quantity of extracellular matrix. That's characteristic of connective tissue, domination of extracellular matrix under the cells, but not ordinary cells. This is a cells lineage, difference type. And what about the fibers? Fibers and ground substance, it's a component of extracellular matrix. And the connective tissue more rich by this fibrous component than other tissue in human bodies. And also, very important functions, participating in supporting homeostasis. Homeostasis is to have to regulate the process of internal environment conditions. And of course, aerobic oxidation process represents without active oxidative reactions. This is connective tissue can be stimulates the process of development. Well, thank you. And now just we talking about what is it, mesenchyme? That's a mesenchyme. Mesenchyme is a multipotential cells of embryo and embryonic connective tissue, as any book is writing. So most connective tissue originate from mesoderm, the middle germ layer of the embryonic tissue. From this layer, the multipotential cells of embryo, mesenchymal cells, developed. Although in certain areas of the head and neck, mesenchyme also develops from neural crest cells of developing embryo and is known as ectomesenchyme. So we just can repeat these materials because 2 weeks ago was discussed some questions about development of embryo and fetus. And just this is moment is very important for us. That's embryonic connective tissue, multipotential cells of embryo, which is named mesenchyme, very active transformate in the different derivatives. Example, one of direction of this development is neural crest cells, which given them ectomesenchyme in the head, in the neck. And also we have the mesenchyme which originates from mesoderm. It's a mesodermic part of mesenchyme. And mesenchyme also originates from entoderm. It's a mesenchyme in the intestinal tract. So you need to remember that the role of mesenchyme in each region of human body to regulation of formation of connective tissue, blood vessels, and any region is also smooth muscle cells. Mesenchymal cells migrate throughout the body, giving the rise to connective tissue. Their cells, including those of bone, cartilage, tendons, capsules, blood, and hematopoietic cells. Next, how is visible that mesenchyme? We have a very good scene again, photo micrograph of microscopic slide of mesenchyme cells. This is hematoxylin and eosin stain, but you have seen that eosin not very active in this photo. Can be identified oxyphilic areas because dominated basophilic structures in cells. It's clearly understood because mesenchyme doesn't contain any fibers. Only cells, extracellular matrix contain only ground substance, but dominated hyaluronic acids, not very developed. Some proteoglycans not can be visible fully, yes, because this is unmature multipotential rudiment of tissue, connective tissue, embryonic connective tissue. Okay, so what about connective tissue like system? Connective tissue, a group of tissues in human body that maintain the form of the body and its organ and provide cohesion and internal support. The connective tissue includes several types of fibrous tissue that vary only in their density and cellularity, as well as the more specialized and recognizable variants like bone, ligaments, tendons, cartilage, and adipose tissue. Also, we have a specialized tissue like reticular connective tissue, elastic connective tissue, and very special mucous connective tissue in umbilical cord. In this slide, we have seen numerous cells and numerous tissues in the different types of connective tissue. First of all, we have seen pluripotent stem cells. This is hematopoietic stem cells, and this is mesenchymic multipotent stem cells. And that's two lineage of the stem cells to give rise to the formation of different differons of cells. Effect of it, we have seen loose connective tissue, reticular connective tissue, dense irregular connective tissue in skin, and tendon like dense irregular connective tissue. It's very good seeing the collagen bundles and loose connective tissue between it. So, also very important, the role of cartilage in human organism, in skeleton, and of course bone. But all that tissues arise from mesenchyme. This is mesenchymic derivatives. And we have seen that's a bone and cartilage at the outside of it have seen the dense connective tissue, which is also named periosteum in bone, perichondrium in cartilage. So structure of connective tissue derivatives very resemble each to other, but has specialized cells and specialized features. Like example in bone, you know that's a extracellular matrix higher mineralized. In the bone, higher hydrate have a lot of free and join water molecules because this is specialized tissues. And the question is, what is a mature connective tissue and what is a specialized connective tissue? Mature connective tissue classified into connective tissue proper and specialized connective tissue. Connective tissue is composed of cells and extracellular matrix. And I was a minute ago tell you about mesenchyme, and was discussed that eosine is not good stain mesenchymic areas because in mesenchyme is absent fibric component, because mesenchyme is not mature connective tissue. But if we talking about differentiation of cells, differentiation of products of cells, we have to see the increasing of oxyphilic or acidophilic component in connective tissue areas. Because that this is extracellular matrix consisting of ground substance and fibers, collagen fibers, reticular fibers, and elastic fibers. Extracellular matrix is dominated in all types of connective tissue. So memorize that. First of all, mature connective tissue characterize very good visible fibrous compartment and cellular compartment. You have to find it with a differential staining, like hematoxylin and eosin stain also can be differentiated. But also we have to know that specialized stain for elastic tissue, specialized stain for reticular tissue is present, and we will discuss and study this moment. Let's now, may I represent to you the types of connective tissue in human body. First of all, this is of course connective tissue proper, which is divided into fibrous connective tissue and specialized connective tissue. Fibrous connective tissue divide into loose irregular connective tissue, shortly LICT, and dense irregular connective tissue, which is divided into dense irregular connective tissue, dermis of skin, and dense regular connective tissue, tendon. So dense connective tissue, very active, represented in human bodies. Like example, except tendon, we know the fascia, you know aponeurosis. So everywhere is present some dense connective tissue. And let's now have talking about specialized connective tissue. One of the very important is skeletal connective tissue, cartilage, bones, dentin, cementum. For all these specialized connective tissue, which is named skeletal, is present the characteristics is higher mineralization of ground substance. Another moment, this is types of connective tissue in organs, like adipose connective tissue, like mucous connective tissue, but that's question we discuss little late at the end of our lecture when special investigates each of these types. Let's now back for connective tissue proper. What is the main base for classification? This is relationship between extracellular matrix and cells. And we have to classify it in fibrous connective tissue. This is fibrous connective tissue, and all another is a characterized special organization. Example, fibrous connective tissue contain a lot of fibers. They can be divided into the loose and dense. Loose connective tissue fibers are situated loosely but contain a lot of ground substance and many types of cells. It's widely distributed in the body and accompanies the blood vessels. With the separation of the loose connective tissue, dense connective tissue, fibers inside this tissue, dense connective, more situated densely situated, and because that dense connective tissue can be divided into regular and irregular. What does it mean? Regular, that's the fibers situated regularly in parallel and to form the bundles, which is in lineage have in lines can be originate from the fibroblast and fibrocyte cells, and we have the example of regular dense connective tissue, tendon, ligaments, fascia. Everywhere we have find the bundles of collagen fibers, bundles of collagen fibers. Irregular connective tissue fibers are situated irregularly in all three directions. And we have like meshworks in dermis reticular layer, in capsule of organs is very good visible. We started our lecture with a slide and word of American writer about the skin, and I was shown to you the dermis reticular layer also in skin. Let's now investigate the localization of cells and vessels inside loose, irregular connective tissue. First of all, why its name irregular? You have seen that collagen fibers and elastic fibers is not regularly located. It's located chaotic. And effect of it, we're talking about loose, irregular connective tissue. From the left side is very good visible arterioles. From the right side we have seen the venule. Between this is arterioles and it's maybe not venule, sorry, maybe it's arteriolaric component of microcirculatory bed and venularic component. But that's shown that loose connective tissue everywhere create the regional structure in organs and to support the vessels for each organ. Okay, now to talking about the cells. Number one, this is a fibroblast cells. Fibroblast cells, the cells with the long processes, is very active cells. Some of fibroblast is resident cells, is not be migrated from another area. And also we have seen adipose cells. Adipose cells accumulates the energetic substances like lipids and phospholipids. Of course, white adipose cells, as we have seen here, also resident cells, is not migrated. But one more, we have seen the plasma cells and macrophage, and that cells is very active for migration because both of these cells arise from blood. Plasma cells arise from lymphocytes. Look at this one, we have seen it here also, lymphocytes. And macrophage arise from monocytes, but also is very good visible number 3. This is mast cells or labrocyte. Mast cells, this is cells of connective tissue which arise, developed from the leukocytic basophils. Because that the mast cells very often is named connective tissue basophils, but correctly named in the histological tradition mast cells or labrocytes. And the last we have seen here the granulocytes. It's maybe neutrophils, maybe eosinophils, but it's not important for us. Now we understood that the cells of blood, especially lymphocytes and leukocytes, very active migrated between blood vessels and connective tissue. In another moment, this is leakage of plasma in the connective tissue. It's a very often take place during regulation of functional condition, not only during disorders and injury. Like example, when some cells deactivated, or maybe when some substances which change the osmotic pressure can be appear in the internal environment, immediately the connective tissues start to regulate homeostasis. And of course, we have seen here the cells, mast cells and eosinophils, which is participate in this process. If you remember our lecture about blood, you can be remember it that both these cells is participate in inflammatory reactions, especially via synthesis of mast cells, histamine and heparin. Today we again talking about it, but I want to tell some words about these fibers. Collagen fibers is more thicker and more spiralized in structure than, for example, tiny elastic fibers. And this is fibers that play the key role in the construction of regional organization in histone. To please writing that histone, this is a morphological unit of regional organization inside an organ. And the one of the component of histone is connective tissue with blood capillaries. Okay, let's talking about functions of connective tissue. Just you have understood what is it. First of all, loose connective tissue provides the exchange of nutrients, metabolites, and waste products between the tissue and circulatory system. Second functions: defense, protection against bacteria. Some connective tissue cells, like part in immune response and phagocytosis, provide immune reactions. Example, macrophage. Example, plasma cells. Example, lymphocytes, which is also migrated inside connective tissue. The supporting function is provided by the presence of numerous collagen and elastic fibers in the intercellular matrix. The dense connective tissue in the dermis or ligaments and tendons play important role like supporting material for organization this structure. Connective tissue take part in the tissue repair, regeneration, reparation. Note that the processes of tissue repair are largely a function of connective tissues. Morphogenetic functions, morphogenetic. Morphogenesis, as you know, this is formation of an organ, shapes and structure of organic composition during time. And for example, the dense connective tissue is involved in the formation of capsule of many organs, loose connective tissue to form trabeculae inside the organs, because the stroma of organs also contain or loose connective tissue or reticular connective tissue in different modifications. So let's now classify connective tissue cells. I was tell you that the first group of cells is named resident cells. The main lineage of the cells is fibroblast, fibrocyte, fibroblast, fibrocyte. Of course, this is a main type of cells in connective tissue. Fibros is a fiber and blastos generator. So the cells will generate a new fibers, fibers of collagen, fibers of elastin, and some other may be reticular fibers. So every time can be dynamically adaptate this tissue for changes. Another type of cells, which is also named adventitial cells, it's originally also arise from stem cells of mesenchyme, pluripotent, multipotent mesenchymic stem cells, because that very often many investigators to talk that adventitial cells not always present, whereas we have find necessary in the red bone marrow, can find, near the vessel we can find, but the disseparate fibroblast cells and adventitial cells only in the structure, very difficult. How we can identify it? Of course, with special cell markers, immunohistochemical methods can be shown it for us. And adipocyte. Adipocyte is also cells which arise from multipotent mesenchymic stem cells. Adipocytes to create adipose tissue, white and brown. Look, but we have seen that macrophage is not joined with resident cells because macrophage is not originally fully resident. Macrophage can be present, one of them is resident, another is not resident, is wandering cells. I was telling you a minute ago also about it. Example, leukocytes, it's wandering cells. Arise from blood, migrated from blood into the connective tissue, and can be back from the connective tissue to blood again. Sometimes this happens, but more seldom. And, of course, plasma cells, mast cells. Plasma cells arise from lymphocytes, mast cells arise from basophils, leukocytic basophils. So this is wandering cells, which is all the time migrated between connective tissue and blood, between blood and connective tissue, but the macrophage located between resident and wandering cells, because one of part of macrophage wandering cells, another is resident, and we have discussed later. Cells of connective tissue, again, some information you need to indicate it in your notes. Fibroblasts are the key cells in connective tissue proper, and fibroblasts originate locally from mesenchymal cells and are permanent resident of connective tissue. And other cells like macrophage, plasma cells, mast cells originate from hematopoietic stem cells in bone marrow, circulate in blood, and then move connective tissue when they function. These and other white blood cells, leukocytes, are transient cells of most connective tissues, where they form various functions for the short period, and after that can be die by apoptosis. Ordinarily, the all blood cells can be dying in the spleen, for example, many. Just talking about cells lineage or cells differome. What is it, cells lineage? A cell lineage is the developmental history of differentiated cells as it traces back to the cells from which it arises. So, developmental history of differentiated cells as it traces back to the cells from which it arises. And the next moment, this is definition of differome, plurality of all types of cells in one lineage of differentiation from stem cells till specialized cells. Again, repeated. Plurality of all types of cells in one lineage of differentiation from stem cells till specialized cells. And we have to see in this interesting scheme or diagram the lineage of cells which participate in immune response and also participate in regulation of homeostasis. Example, lymphoid lineage, myeloid lineage, and connective tissue lineage. You will discuss that question in the hematopoiesis, but look at this. Hematopoietic stem cells arise from mesoderm, multipotent stem cells, and this is multipotent mesenchymic stem cells transforming into the mesenchymal stem cells and to hematopoietic stem cells. Both of them is mesodermal region. First is lineage of lineage will be common lymphoid progenitor. So stem cells give us a formation of progenitor cells. Next progenitor cells, T cells, will be the some precursor cells. Blast. He is not be identified, but we have to talking about lymphoblast, B, T, and some other. And another one variant, this is myeloid lineage, myeloid progenitor cells. This is progenitor cells give rise to formation of erythroid line, like megakaryocyte and erythroid progenitor. Also we have erythrocytes, red blood cells, erythroblastic lineage, and megakaryocyte, this is platelets formation. And all another is macrophage progenitor cells. Look, from pre-monocytes, this is precursor of monocytes, to can be developed dendritic cells, osteoclast in bone, macrophage, classical, and granulocytes is also can be developed from myeloid line. It's not macrophage line, but it's common origin only. And we just back for our connective tissue. Look, mesenchymal stem cells can be give rise to formation of osteoblast. This is a future bone. Adipocyte. This is a future adipose tissue. Chondroblast. This is a future cartilage. And the myoblast, like smooth muscle cells, progenitor. That's transformation of cells regulated by genetical factors. And we have to writing the definition of two important terminological process, maybe morphological process, sorry. That's the first of all is determination. In the previous classes, previous lectures about the embryology, we was writing that development proceeds in the series of stages in which precursor cells are first restricted in their developmental potential, determination, and subsequently express their genetic information as a specific tissue, differentiation. Let's now can be define this term, that determination is restriction of cells ability for transformation in different lineage and expression of genetic information as specialized lineage of cells and tissue. Differentiation, the process by which cells become progressively more specialized, and normal process through which cells mature. This process of specialization for the cells comes at the expense of its breadth of potential. So, again, about determination for discuss each moment. Restriction. Restriction, this is decreased potential for development. And the cell ability for transformation in different lineage. And in this, we have about talking some potential for development like totipotency in blastomeres after beginning of cleavage in embryo. Also, we know about pluripotency, like example, pluripotent stem cells in the hematopoietic stem cells. In this case, many types of cells can appear, but is only cells but not tissues. And multipotent cells like mesenchymic cells. Finally, we have talking about precursor cells like progenitor cells also. This will be blast cells like lymphoblast, like for example fibroblast and some other. In this case, with each process of determination can be present like restriction of ability of specialization. Effect of this will be the formation of specialized lineage of cells in tissue. Differentiation. This is specialization of cells. Progressively more specialized cells, and this is process to provide the realization of all potential, but breathe of this potential inside one lineage of cells. Okay, let's now talking about mesenchymal stem cells. To have seen how is multipotent mesenchymal stem cells give us the formation of different cell difference. First type of cell difference will be osteoblastic cell difference. This is formation of bone, osteoblast, osteocytes, and the stages: proliferation, lineage progression, multiplication, differentiation, maturation. Look at this, we have seen how the cells embedding itself inside the mineralized matrix. Next, chondrogenesis. In chondrogenesis, the formation of cartilage, we have also chondrocytes, but the chondrocytes arise from chondroblast, prechondroblast, next chondroblast, and we have also seen hypertrophic reaction surround the chondrocytes mature. It will be also maturated via the formation of new proteins of extracellular matrix. And in this case, we're talking about chondromucoid in cartilage. In the bone, we have talking about osteomucoid, like extracellular matrix. Well, myogenesis. Myogenesis we will discuss in the next topic about the muscle tissue, but it also can be arise from mesenchymic cell, and also we have seen the bone marrow cells, which to give rise to formation of reticular stromal cells, reticular cells. And the main line of differentiation in this schematic diagram, this is tendinogenesis, ligamentogenesis, the formation of tendon and ligaments. As you have seen, again, we have visible transitory fibroblast cells. This is transitory fibroblast cells can be migrated for any area where it's to form in new ligaments or maybe tendons. And of course, mature cells is named fibrocyte. Sometimes for tendon is named tendinocytes. Adipogenesis, the formation of fatty tissue. And we have seen here the pre-adipocyte, early adipocytes, adipocytes of multi-vacuoles. This is brown adipose cells, and adipocytes with a big one vacuoles. This is a classical white adipose cells. So this is transformation is visible like result of differentiation after special determination. So talking about fibroblast. Look, we have seen in this photo fluorescent confocal microscopy of fibroblast cells. Inside fibroblast cells is very good visible not only nucleus, but also we have seen numerous microtubules and also very good visible cytoplasmic intermediate filaments. For connective tissue and mesenchyme, the main type of intermediate filaments vimentin. But microtubules as well, everywhere is tubulins containing structure. But vimentin you need to remember. And also we have seen here the ordinary light microscopic microphoto under hematoxylin and eosin staining. So what about fibroblast? Also can be seen in this new slide. We have very interesting electron microscopic photo, transmissional electron microscopy. In this photo we have seen collagen protofibrils. Is not a full collagen fibrils, it's a protofibrils, is accumulated, like here is also visible. And we also have seen the nucleus. Nucleus has a lot of yards, nuclearic yards, and also have very good visible heterochromatin, nucleolus, euchromatin, and very good developed component of membranous organelles, especially rough and smooth endoplasmic reticulum. And in this schematic diagram is visible like a fibroblast to change the shape and also can synthesize the different type of collagen fibers. It started from the tropocollagen molecules, collagen protofibrils, protofibril, and after that bundles of collagens fiber. We have to talk about structure and functions. That's the fibroblast is a spindle shape of stellate cells. Its nucleus is round with a lot of euchromatin, active chromatin, well developed endoplasmic reticulum, which is collagen synthesized and possible to synthesize produced elastin or reticular fibers is collagen type 3, as well as protoglycans and glycoproteins for ground substance are synthesized. Fibroblast produces the extracellular matrix constantly. It is a reason why the border lines of fibroblast under the light microscope are indistinct. Look at this one, we have seen that the line is indistinct because produced a lot of fibers. This is a photo of electron photomicrograph examination micrograph in your exam will be. Look, this is a nucleus. Nucleus and heterochromatin. Heterochromatin at the periphery with nuclear pore is also visible. Also, we have seen a very good developed rough endoplasmic reticulum. This is a gynous and a lot of ribosomes, polysomes here, especially polysomes. And the free ribosomes is also visible in the cytoplasm, separate from reticulum. Also visible mitochondria, Golgi complex, and we have seen that the protofibrils of collagen in different levels of organization can be seen surround the fibroblast. Fibroblasts, the most common cells in connective tissue proper, maintain most of tissue's extracellular components. Fibroblasts synthesize and secrete collagen, elastin, and glycosaminoglycans, GAGs, proteoglycans, and multi-adhesive glycoproteins that comprise the ground substance. As described later, the most of the secreted extracellular matrix components undergo further modification outside the cells before assembling as a matrix. Second type of cells in the proper connective tissue, this is myofibroblast. Myofibroblast, of course, reagates on some factors, proteins called the growth factors that influence the cell's growth and differentiation. In adults, this is results of proliferation of fibroblast, especially junior fibroblast, can be transforming into the myofibroblast. If we have seen, myofibroblast play the key role in wound healing near the injuria in the skin, sometimes called myofibroblast, and well-developed contractile functions. We have seen here the bundles of contractile myofilaments in the cytoplasm. Also have seen the very big vacuoles with digestion substances. This is phagocytotical vacuoles. And also we have to know that these cells can be replacement. The smooth muscle cells increase the diameter of an organ. Like example, hyperplasia of uterus. Upper picture, look, this is uterus, and in uterus during the pregnancy of female, diameter of wall increase in more than 10 times, and this is a moment of hyperplasia, of increasing of the quantities of cells via proliferation of myofibroblast, not smooth muscle cells, but myofibroblast. And this is myofibroblast very fast to degenerate after the pregnancy. Also a moment, this is wound healing. That's myofibroblast can be stretched the area of destruction of the skin, dermis and epidermis, and especially dermis. Well, so ground substance organization. Let's now talking about elements of ground substance. It consists from glycosaminoglycans, GAGs. The one of the famous is hyaluronic acids, and another, this is chondroitin 4-sulfate, chondroitin 6-sulfate, dermatan sulfate, heparan sulfate. This is glycosaminoglycans is represented for special type of tissues. Like example, chondroitin sulfate dominated in the cartilage, or dermatan sulfate ordinarily present in the dermis, connective tissue of dermis of skin. But we have talking about proteoglycans. Proteoglycans is protein core to which glycosaminoglycans covalently bound aglycan, syndican, and fibroglycan. That is very good visible in scheme. Look, this is a blue color line, maybe blue color core. This is hyaluronic acid. Also we have seen it, this one. From another side, we have seen how this round small particles, this is joined proteins, to join the or core proteins, to join the glycosaminoglycans with proteoglycans. And proteoglycans is visible here, surround the proteoglycans. So, hyaluronic acids, glycosaminoglycans green color, and proteoglycans red color. Hyaluronic acid, the main core, and the small branch, this is glycosaminoglycans, and proteoglycans, small sub-branching. We have seen how this structure is joined with collagen fibers. This is big, thick structure is collagen, purple color, and joined with cells through the fibronectin, special proteins for junction. Glycoproteins, fibronectin, laminin, osteonectin, osteopantin, chondronectin. So, let's now you understood that some cells all cells need to interact with extracellular matrix fluid, but it's not a fluid, it's a special matrix because it's a structural organized. The main type of it is, of course, collagen fibers which embedded inside ground substance. But the collagen fibers is surrounding by glycosaminoglycans subunit, as we have seen it here. And glycosaminoglycans joined with proteoglycans, and this is special molecules which to provide the hydration of this extracellular matrix, and through the glycoproteins like fibronectin, like laminin, like osteopantin and chondronectin to join the cell with fibers. Next table to show for us the composition and distribution of glycosaminoglycans in connective tissue and their interactions with collagen fibers. Hyaluronic acid and glucosamine to join with ordinary umbilical cord, synovial fluid, vitreous humor, and cartilage. Of course, this is very important for these organs. Chondroitin sulfate to join with galactosamine in the cartilage, bone, cornea, skin, notochord, aorta, and to provide the higher level of interactions, mainly with collagen type 2. Chondroitin sulfate 6, it's joined with glucuronic acid and galactosamine in cartilage, in umbilical cord, in skin, in aorta, media of aorta, and to provide the high levels of interaction, and again with collagen type 2. Dermatan sulfate aggregates with iodonic acid and glucuronic acid, and also galactosamine. Is represented dermatan sulfate in skin, tendon, aorta, and adventitia. Heparan sulfate to represent it in the aorta, lung, liver, basal lamina of epithelium, and intermediate levels of interaction between collagen type 3 and collagen type 4. as you have to, we little forget that the dermatan sulfate provide the interactions with the collagen type 2, type 1 dermatan sulfate. Keratan sulfate, again represented in the cartilage. And that's minimum that you need to know. How is visible under the electron microscope? Under the electron microscopic transmissional electron microscopic technique, we have seen glycosaminoglycans and proteoglycans, like this is aggregations of substances. It's very high magnification. So this is photo to confirm that glycosaminoglycans is not ordinary fluid. This is a structural proteoglycans and glycosaminoglycans complex. In ground substance of cartilage, also have been seen the analogous of what is named chondra mucoid, or the main substance of cartilage. We have seen here the isogenic group of chondrocytes, ordinary 4 or 2, and we have seen here hyaluronic acid, again green color, and this is a fibrous of collagen. Okay, next, fibronectin. Fibronectin, how is visible under the microscope? Under the microscope, we have to identify fibronectin with immunohistochemical methods. This is a red color area, intensive red color, to show the cells, maybe special basal lamina in the tissue, which join with some cells. Look, this is epithelial cells, as I think. Yes, and we have seen the intensivity of this is red color to show where is the fibronectin more active. In the upper side, in the middle side not, in this side from right portion, here also, but is not visible in the center. So this is a protein which provides a junction between cells and cells matrix, especially with collagen fibers. And this system is very dynamical, adaptate during the time. So let's talking about collagen fibers. The collagen fibers are the most abundant type of connective tissue fiber. They are flexible and have a remarkably high tensile strength. In the light microscope, collagen fibers typically appears as a wave structure of variable weight and indeterminable length. They also can be stained by acidic dyes and coloring by, for example, aniline blue. Also can be identified with Mallory connective tissue stain, and Masson trichromatic stain can be light green. So in future I have shown to you, this is photomicroscopic micrograph, but just talking about electron microscopic, more important. So under the transmissional electron microscope, we have to identify this interesting structure and especially collagen fibrils. With individual fiber, the collagen fibrils are relatively uniform in diameter, in different locations and a different stage of development. However, the fibers differ in size. In developing of immature tissue, the fibrils may be as small as 15-20 nanometers in diameter. In dense regular connective tissue of tendons or other tissues that are the subject to considerable stress, they may measure it of 300 nanometers in diameter. This is a dark line of protofibrils. This is protofibrils, collagen protofibrils. Light line and 3 is special protofibrils. So, well, where is it? What's the type of collagen? Because there's a difference relationship between organization of collagen molecules is present. First of all, this is collagen type 1, a diameter of molecule 300 nanometers, 67 nanometers bonded fibrils, thick and highly picroserous to identified, and also we have seen in the skin, tendon, bone, and dentin. And of course functions is resistance to tension. Collagen type 2. Collagen type 2, also 300 nanometers, same parameters. Loose aggregates of fibrils, by fingering, and also we have the fingering end. Yes, and also we have the cartilage, vitreous body, and provide resistance to pressure. The next is collagen type 3, and 67 nanometers banded fibrils, ordinary thin, weakly bare fringent, and also we have the identified with silver impregnation sometimes, argyrophilic. Type 3 is also named like argyrophilic fiber or reticular connective fiber. And also we have to see that collagen type 3 is present in skin, muscle, blood vessels, and aggregates with the collagen type 1. Structural maintenance in expansible organs. Collagen type 5. Diameter is 390 nanometers to provide the terminal globular domain, and also the present in fetal tissue, skin, bone, placenta, and interstitial tissues of internal organ. Also participates in the junction with the collagen type 1. 9, 11, and 6 type of collagen we have discussed in the practical classes. Like example, the type 4 collagen is present in basal lamina, in epithelial tissue also. Well, let's now talking about molecular organization. Molecular organization, that's collagen fibers have 68 nanometers bonding pattern. The collagen molecule, formerly called tropocollagen, measured about 300 nanometers long and 1.5 nanometers thick in the head and tail. Each collagen molecule is a triple helix composed of the 3 intertwined polypeptide chains. Amino acid in the chain is a glycine molecule and hydroproline molecule, or hydrolysine and proline molecule. All the time is covalented, not covalented, but with a joined each to other during the polymerization. And of course collagen synthesis and degradation of. We have to immediately migrate for the description of how is collagen synthesized. The formation of collagen fibers involves the uptake of amino acids, proline, lysine, etc. by fibroblasts, the production of polypeptide chains in rough endoplasmic reticulum inside dead cells. And modifications of polypeptide chains occurs with the cisterna of rough endoplasmic reticulum and Golgi complex. The resultant molecules are called procollagen. And this is procollagen packing into the secretory vesicles and leaving the cells by exocytosis. Like example, I just shown to you, this moment is visible here. This is a formation of this protocollagen molecules, and through the with parallel with glycosaminoglycans is also synthesized and migrated into the outside intercellular area. Back now for our talking. So, steps of collagen production. The protocollagen chains are produced on polyribosomes on rough endoplasmic reticulum, translocated into the cisterna. These typically have long central domains, rich as proline and lysine. In type 1 collagen, every third amino acid is glycine in the composition. The hydrolyse enzymes in endoplasmic reticulum cisterna edit the hydroxyl and the groups of some prolines and lysines. Reactions of inquire the ascorbic acid, vitamin C, as cofactors. So that's very important to remember. That's the vitamin C ascorbic acid, participate in the biosynthesis of collagen molecules. Glycosylation of hydrolysine reduces also occurs in different degrees type of various collagen types. As effect of that, we have seen this is schematic diagram how the collagen synthesized. Golgi apparatus, packaging and secretion of procollagen. Next is the procollagen translocate into aggregates with some hydroproline to form the tropocollagen. Next, collagen fibrils levels. And next, collagen fiber. Collagen fiber because we have seen how it forms the bundles of collagen fibrils under the activations with proteoglycans and glycosaminoglycans. So next moment, assembly of collagen. Assembly of collagen, it's a type of stages of maturation. Tropocollagen molecules, 280 nanometers. Collagen protofibrils, collagen fibers, and bundles of collagen fibers. Again, have seen it here, but we need to continue our talks. This is a collagen fiber. Each of them is contained, this is bundle of collagen fiber. Each is contained collagen fiber. Each collagen fiber contain collagen fibril, and collagen fibril contain protofibrils. Okay, next moment. Collagen degradation. Collagen degradation can be visible under microscope if we have to investigate some areas of after the remodulation, after the injury, like for example, ordinary take place activities of macrophage and fibroclast specialized cells for destruction of connective tissue. And the main enzymes is metalloproteinase, collagenase, hyaluronidase. What about reticular fibers? Reticular fibers is collagen type 3, we was discussed that. But that reticular fibers to form the meshwork of stroma for hematopoietic organs, ordinary. And we have to see this is reticular connective tissue, which is very good identified in the stroma of lymph nodes. We have seen here lymphocytes also at periphery, but in the center is very good visible reticular fibers with reticular cells, which like to form the meshwork for maturation and housing of lymphoid tissue. Elastic fibers. Elastic fibers are elastic. They can stretch after distension. Elastic fibers are thinner than collagen fibers. They can also comprise bundles but can branching. They are composed of elastin core and microfibrous sheath. Elastic is synthesized by the same pathway of collagen. Look at this one. We have seen again fibroblast. We have seen the package of Golgi apparatus. We have the special assemblage of single elastic fibers. And we have seen fibrillin, fibrillins, tropoelastin, which like middle elements for the aggregations of bundles of elastic fibers. Sometimes we have seen, sometimes it's named oxytalan and L-adin fibers, which is not mature. And here, very good visible under the scanning electron microscope, the collagen and elastic fibers relations in the connective tissue. In pathology, we have the sometimes disorders in molecular structure of collagen. In this case, we are talking about Ehlers-Danlos syndrome is visible in this photo. It's mutation, special mutation of genes. But maybe you have talking about in the biology, but now that's ideas, that's Ehlers-Danlos syndrome is a result of reach of type of collagen 1 under the another type of collagens. And also we have seen the Strickler syndrome, also specialized disorders. All these syndromes, like also Marfan syndrome, you have seen the famous American president Abraham Lincoln was been suffering on the Marfan syndrome, but the etiology, it's a fibrillin proteins encoding. So this is fibrillin disorders is provide the problem with elastic fibers in aorta. And if, for example, talking about the Marfan syndrome, it's arachnodactylia, also mitral valve prolapse, and dilation root of aorta, aortic dissection can be shown, this is syndrome, and very more of these people is dying very in young time, but not always. And of course, macrophages. Now they're talking about the special type of cells, very shortly discussed, that is the functions of another cells in connective tissue. Macrophages, the big eaters, can phagocyte, catch the biggest bacteria and foreign particles. Macrophages are phagocytotic cells derived from monocytes. Therefore, they contain mainly lysosomes and phagolysosomes as well as the other organelles. They can actively model intercellular matrix and contain winding borderline and because of numerous folds of finger-like processes, which is like a variant of pseudopodia. And also these cells provide the antigen presenting function. This is antigen presenting cells, and they are processing and presentation of foreign particles, antigens on the surfaces for activations of T helpers and T killer cells. They are derived from the blood monocytes and migrated into the connective tissue after differentiation. Acid phosphatase. This is a main marker for histochemistry, histochemical identification of macrophages. Acid phosphatase. Lysosomes are abundant in the cytoplasm and can be revelated by staining for acid phosphatase activities, both of light microscope and with the transmissional electron microscope. The positive reaction further aid in identification of macrophage. And we have the scene, this is macrophage in this photo. This is a nucleus. This is a finger-like projection of pseudopodia. We also have seen the numerous lysosomes, primary and secondary, and phagolysosomes also. And also we have seen lamellapodius is sometimes his name, and mitochondria also can be seen. Next, this is different activities. This is active and non-active macrophage. This is macrophage non-active, and that macrophage is active. How we identify? Because increase the quantities of lysosomes increasing, and we have to calculate it, how many lysosomes here visible. And also we have seen the system of mononuclear phagocytes. This is a system was been discovered by famous Russian histologist Mechnikov. That's the resident cells. We only named it. Langerhans cells in epidermis, dendritic cells in the lymph nodes, Hofbauer cells in the placenta, Kupffer cells in the liver, osteoclast in the bone, microglial cells in the brain, intestinal macrophage is also specialized, but is also these residental cells. Is also we have seen the interstitial macrophage. Interstitial macrophage is present in the lung, for example. One macrophage in lung interstitial, another is dust cells. Look, but absolutely another classification. That's resident cells not migrated outside the organ, but wandering cells can be migrated, like histiocytes. This is a macrophage of the loose connective tissue. Serospaced cells. This is cells which to provide protection against inflammations, and also dust cells. We also have, talking about macrophage in the lung, exudative macrophage. During the big exudative processes can be disintegrate the destruction. And also example, this is macrophage of Kupffer cells in the liver. This is a liver lobule. And we have seen the hepatocytes, and with a special Indian ink, this is special stain for macrophages. We have identified the macrophages in the wall of space of descent. Plasma cells, they derive from B lymphocytes. Plasma cells contain well-developed rough endoplasmic reticulum, the reason for high basophilia of cytoplasm. Plasma cells produce immunoglobulins, antibodies, and responsible for humoral immunity. We have seen that Golgi region with a high macula near the nucleus, and the nucleus of plasma cells resemble on the wheel of bicycle, because we have the special heterochromatin spots resemble on the bicycle wheel. And also we have seen this polarity, that's a nucleus in one area, but rough endoplasmic reticulum and Golgi complex in another area. And all this you need to remember, because this is a examination microgram for you. Mast cells was being identified primary Paul Ehrlich in 1878, and mast cells characterize the metachromatic granules. So that's if we have to use toluidine blue, like here we have seen that the nucleus is a blue color, but granules it's pink blue color. So this is metachromatic effect. That's mast cells, tissue basophils. As a rule, mast cells are located around the small blood vessels. They contain and release the big granules with the heparin, histamine, and other mediators that initiate the inflammatory and allergic reaction. especially histamine to increase the ability of leakage of plasma outside the vessel and increase the diameter of vessels. Heparin stop the hemocoagulation and start activate the blood reach of the some areas. And also we have talking about allergic reaction mediated through the activities of immunoglobulin E on the surface of mast cells. They are produced by plasma cells and because the triggers for degranulation. We have seen it now. Look, the mast cells outer structure. We have seen the big round nuclei, and this is nuclei to provide the functions of, we have seen the higher biosynthesis, and we also have seen the many granules in the cytoplasm, many granules. And so mitochondria is also visible here, and also visible protrusions from outside. And the degranulation of mast cells is also have been identified. So this is mast cells not degranulate, this is during degranulation. And during the junction with immunoglobulin E, antigens like allergens can be initiate the through adenylate cyclase reaction for the degranulation via the activities of calcium ions. And this is granules release heparin, histamine, proteoglycans, and factors of activities of eosinophils. As a effect of that, we have identified this is mast cells near the vessels, around the vessels. The mast cells provide regulation of regional homeostasis. And we have to see it, that's leukocytic infiltration in connective tissue during inflammation. So this is during inflammation, the blood plasma is leakage outside and the cells also migrated. This is a plasmargia, and this is a leukocytic infiltration. We have seen here different types of blood cells, also eosinophils and neutrophils is visible. Also can be seen that's now dense connective tissue very shortly. Irregular fibers are situated in going the three directions. So this is a dense, irregular connective tissue. And we have seen fibroblast everywhere is visible. Also tendon. The tendon consists of bundles of type 1 collagen fibers surrounded by fibrosides. And several bundles of type 1 are combined into the bundles of type 2 surrounding the loose connective tissue layer endothelium. And the endotendinium. And also we have to know that several bundles of type 2 combine into the bundles type 3. And we have another variant of thickening loose connective tissue layer, peritendinium. And outside is visible like a layer of epitendinium. We have seen it here. This is endotendinium between epitendinium outside and epitendinium that's our outmost the structure. And we have also contained the few cells, the fibrosides, the main dominated in these structures, package fibers, regular fibers situated regularly in parallel, and the tendons, ligaments, and cornea characterized. Special connective tissue, we was talking about reticular connective tissue, now talking about pigment cells. Pigment cells, melanocytes, developed and arise from the nasal crest. This is unique specificity during all the other connective tissue cells, and these cells contain a lot of granules of pigment in the cytoplasm and have stellate shape. Adipose tissue. The adipose tissue is divided in two types, white and brown. White adipose tissue is composed of large fat cells of adipocytes, and this is type of cells we can be identified here. This is a white adipose cells with a big vacuoles in the center, diameter 30-50 micrometers. In structure and functions, the fat cells organize in group of called lobules, and lobules of fat, and separate loose connective tissue by septa, which is blood and nervous adipose tissue. At the end of our lecture, we have seen that's a position, localization of the adipose tissue, white adipose cells and brown adipose cells, and we have seen the structure of brown adipose cell, which is more effectively with specialized granules, which is located in the center of cells, not like ordinary have seen, not like the white adipose cells, where is big vacuoles with phospholipids. And main function is storage, ordinary energy production, adipose tissue in hypodermis, mesentery, ameta, and retroperitoneum. We have also talks about the functions of adipose tissue, reserve of calorie materials, fat storage, thermoisolation, heat insulator, story of water, replacement of involving organs, thymus, red bone marrow, mammary gland, elastic pad in palms and soles, mechanical support around the kidney. And brown adipose tissue, we have talking only for newborns and sometimes in area which is a small, provide the higher production of ATP. In the body, in humans, we have two amount of newborns and also located posterior triangle of neck around the thyroid glands, carotid arteries, and some other. And mucous connective tissue, this is connective tissue of umbilical cord, contain the fibroblast-like cells. We have seen it like fibroblast, but very density, and extracellular matrix. Fibers not very good developed in this case, and this is like specialized tissue for prevent the destruction and pressing of vessels in umbilical cord. Thank you very much. Our lecture is finished. Next lecture will be after 2 weeks. Goodbye, students.